Good morning. Multicellularity. I just want to use a few minutes of time to talk about how, and when multicellularity evolved, focusing on eukaryotes. And I just want to draw a few major themes out for you from the John Bonner article that you're all reading this week. First off, there are many origins of multicellularity, and I'll show you some of them across the tree in just a minute. Second, that there are these trends in aquatic versus terrestrial. So look for that in the Bonner article, and I'll talk about that here. And finally, there is a difference between thinking about why something originated and why something has been maintained. So becoming multicellularity allows organisms to get large and down the road to differentiate different tissues, although there are differentiated single-celled organisms as well. But we have to think about it at the moment of origin of multicellularity. The organisms weren't thinking, oh, down the road, I'll differentiate into many tissues. So maybe multicellularity has been maintained for these differentiation reasons, but don't confuse that with origins. So how many times has multicellularity evolved in eukaryotes? I don't know. Last time I counted 20, 25, 30 times. Here's a phylogeny. We've seen versions of this all semester. And we see multicellularity, or at least multinucleated large cells in forams, diatoms, brown algae, which are the kelps, the labyrinthalids, slime nets, I'll tell you about them today, ciliates, red algae, green algae, myxogastrids, or the acellular slimal, dictystellids, or the cellular slimals, animals, choanoflagellates, fungi, and many, many more. I'm not going to go over all of these, but just a few, and I'm going to start with ciliates because they're the highest life form out there. So ciliates have become multicellular many times in both aquatic and terrestrial ecosystems. And here we'll see that difference that Bonner picks up as a major theme. In aquatic systems like this, you have organisms that divide and fail to separate. So this entire colony here is colonial. It started as one cell, and then the cell divided into two, and then each cell divides and divides. So these two are two recent sister cells from a mitotic division, and there are other pairs in here. So in aquatic multicelled ciliates, you divide without separating. This is Zoothamnium, which is like Vorticella, in the same group as Vorticella, and we saw Vorticella several times in lab. Here's another picture of these clonal ciliates. Ciliates have also become multicellular on land. This is Soragina, which an undergrad at Smith wrote a paper on quite a few years ago, doing the molecular phylogeny. And this is a ciliate that lives in the soil, and when the food runs out, it aggregates into a little mound, and then it actually excretes this stalk. And so each ball up here is an individual ciliate in a resting stage or cyst waiting. So these guys in the soil in terrestrial ecosystem aggregate to this multicell stage, and this is perhaps a dispersal structure to enable the ciliates to get out to other areas where there might be more food. Another example of a multicelled organism that we haven't met yet are the labyrinthalids. I think I mentioned them briefly when I talked about straminopiles, which are part of SAR. These crazy organisms actually build highways that they travel within. So what they look like here, there are individual amoebae, and they excrete a highway. So this is the scanning electron micrograph of the highway. And here they are in their highway, and they've somebody's added yeast to this culture, and they're streaming in to feed on the yeast. So I leave it to you, but whether you call this some stage, transitional stage to multicellular, truly multicelled, but it is a highway network with many cells living within it, certainly colonial, and I would argue on the scale of multicellularity. The next group to highlight are the dictystellids, which are the cellular slime molds. These can be an amoeba, they can be a slug, or they can form a fruiting body. They're extremely common in soils. So here you can, see a single-celled amoeba. They can aggregate to a multicelled slug, and then they can sit down and they make a fruiting body, although here, instead of excreting a stalk, some cells differentiate into stalk and give up they sacrifice themselves to lift these spores up into the air to start future generations. The life cycle you'll see in the John Bonner movie, or you already saw in the John Bonner movie. You start with amoeba. They aggregate together to form these mounds that then turn into these slugs that crawl around and then differentiate into fruiting bodies. And they're so beautiful, put in another image. So here it is going from slug to fruiting body. And again, these are spores and these are cells that have differentiated into stalk and they die, they leave no offspring. These fruiting bodies can be very simple, like in Dictyostelium discoideum. They can be more complex in Dictystelia polycephalum, or they can be quite complicated like Polyspondylium here. Beautiful organisms. Another example is Physarum, the so-called acellular slime molds. These are amoeba. They can be biflagellate, so I'm not sure why they ever were in Unicont, but that's another story. And they make a multinucleated structure called a plasmodium, confusingly, and they make fruiting bodies. And this picture is, these are oat flakes, and this gigantic mass of slimy stuff is multinucleated tissue. So you have mitosis without cytokinesis, and you get these gigantic slime molds. They can be 3 meters in length. Here is another slime mold growing on a pine branch, just all one cell here, many, many nuclei. Here is a little fruiting body. These guys also differentiate stalk and the spores at the top for my dispersal. Here's another beautiful one. Its body looks like this one, multinucleated, and here are its fruiting bodies. Another is in the green algae. Green algae, I think within them have 3 or 4 or 5 origins of multicellularity, of which land plants are only one. These vulvakine green algae can exist in very simple forms like Chlamydomonas, a single cell with 2 flagella, and then they can go through 4-stage, 8, 16, all the way up to these complex multicelled stages. And of course, people argued for a continuum from unicell to multicellularity in a line, but you guys know better. So just to show you some of these stages here, so this is I think this is Euderina or Pandorina, so it's multicelled, has flagella on the outside, but it is not as complex as the genus Volvox. Each of these looks like an individual Chlamydomonas embedded in a matrix with reproductive cells inside. Clear example of multicellularity with development. So here is the mother colony. These are reproductive structures inside. And here, when you look up close, each one of these is a single cell like a Chlamydomonas. And here are these Volvox colonies. They have male reproductive structures, female reproductive structures, and they have daughter colonies that grow within these multicelled colonies. So again, the argument is always in evolution, before you become an evolutionary biologist, I think the simple default is to imagine everything went from simple to complex, and of course not. But we'll watch just 2 minutes of a video on the life cycle of Volvox.
The ultimate expression of Volvocalian evolution is Volvox, whose beautiful colonies are large enough to be visible with the naked eye. The eye of the needle gives a sense of scale. Each colony is a sphere consisting of hundreds of tiny somatic cells and a few much larger reproductive cells called gonidia. Most gonidia in this sample have already started forming embryos, so individuals may contain several generations of colonies. Starting with this 2-celled embryo, we can see how the gonidia arise. During one particular cycle of division, certain cells divide unequally. A couple are indicated by arrows. The larger cells thus arising become the new gonidia, and they lie at first on the outside of the embryo. They stop dividing, while the somatic cells continue synchronized divisions until the full complement of cells is reached. Inversion takes about 40 minutes. Once the embryos have inverted and enlarged.
All right, so we will stop there. And for those of you who've taken development, that certainly looked a lot like gastrulation. complex development in a, I don't know, microbial eukaryote. So again, linear to complicated. I've posted these slides in our shared Google Drive, and you can take a look if you want to learn more about their life cycle. But what I want to just show is a out of date and yet still accurate. I use it because it's simple. Here is Chlamydomonas, the single cell member of this group, which is also polyphyletic, the genus Chlamydomonas. And we have things like Euderina and Pandorina, which are less complicated than the genus Volvox. But if we focus just in on the genus Volvox, we find it here, we find it in these three places, and we find it here. And so the conclusion from this would be that the genus Volvox and this complex multi-celled life cycle has likely arisen many times within the Volvoquine algae. I'll end with this last example of choanoflagellates, as they are sister to animals. And I will talk about them in a separate lecture. Choanoflagellates have this beautiful basket collar with a single flagellum. They're an episthoconch. But today, I want you to know they can be solitary or colonial. They can also be stalked or stalkless. And the reason why we know they're sister to animals is based on molecular phylogeny and morphology. Here is one of these, an SEM of a single choanoflagellate. Here's its basket, and the flagellum would emerge from here. These are bacteria on the cell. Here's a colonial. Now we get into multicelled. So it's also an aquatic origins, multicellularity, divide without separating. But they can also form these colonies in which they have differentiation. So you have feeding cells on the outside, amoebocytes, and reproductive structures on the inside. And these cells can travel through this matrix and differentiate. So they're like stem cells. They can change their fate throughout this body. And you might think this is very weird. Oh, here's an SEM of these single cell choanoflagellate and a colonial choanoflagellate. But if you've taken invertebrate biology, you might know that these choanoflagellate colonies are a whole lot like a sponge. So this is a, quote, true animal. It's a sponge. It also has these choanocytes inside of it for feeding, and it also can differentiate as these cells move around. And if you take a sponge and put it through a sieve, the cells can reaggregate and redifferentiate into this multicelled body. So a pretty cool connection between choanoflagellates and sponges. So I told you about many origins. I highlighted some of the aquatic versus terrestrial differences. I argued at the beginning that when we think about why multicellularity originated, we shouldn't get confused with why it might have been maintained or allowed to expand. And that is it for today. So thank you.